All right, this is going to be the first of a series of videos that cover the Science 10 Biology unit at Center High campus. I'm going to work through the workbook. I'm going to divide the workbook into 5 sections. If you're currently enrolled in Science 10 at Center High campus, then the pacing might be different than what you're doing in class. Likely, some of the lessons that I'm going to run through in this YouTube video playlist are going to encompass more than one lesson in your class. So if you're using this to review the lesson that you just were part of, then great work, first of all, on doing some review. But feel free to pause the video and then return to it after you finish this classwork in class, or you can be using this to catch up, or perhaps you're learning the whole unit from these videos. This first lesson here, we're going to talk about cells, the first 2 lessons actually, and the idea of what makes a living thing a living thing. The study of cells is called cytology, and you're going to learn in these series of lessons that both animals and plants, and in fact all living things, are made of cells. And you're going to be able to describe the difference between prokaryotic cells, which are more simple, and eukaryotic cells that have more types of organelles within them. And you're going to be able to describe the function of all of those different organelles and be able to relate the differences and similarities between plant and animal cells. Here's the first key thing. All living things are made of cells. Living things, which I'm going to highlight the word living in green here. Living things, another word that we might use for living things, are biotic. And a word that we might use for non-living things is abiotic, which I'll highlight in red here. Instead of being made up of cells, abiotic things are only made up of atoms. I'm going to highlight the word only there because I don't want you to get confused and think that living things aren't made of atoms. It's just that their cells are made of atoms. Abiotic things don't have cells. Biotic is also known as living. We're going to write that here. And you're going to see this prefix bio quite a bit. I mean, we're in the biology unit. Bio means life. So if you go about science classes and you read words that have the bio prefix, like biochemistry, that's the chemistry of life. Biology is the study of living things. Conversely, abiotic things are non-living. The meaning of the prefix bio hasn't changed. Bio is still life, but A is not, or that's how we're going to treat it. Maybe you're taking a class that's synchronous, meaning that you all meet at the same time, but maybe in the past you've taken a class that's asynchronous, meaning that you don't all meet at the same time. By adding this A at the beginning of the word, then we've made it what we call an antonym, or the opposite of this word on this side. So you can think of lots of examples of living things. People are living. Plants are living. All sorts of animals. You could put examples of different animals there, like birds or whales or dogs, insects. But there's also some very small living things, like bacteria are living. Or fungi are living. Things like a mold or a mushroom. Those are living things. Abiotic things are not living. We'll look at the characteristics that living things have in a bit, and you'll see that all of the things I'm about to list here don't have those characteristics. Things that are not living would be things like water. Water is made up of the atoms, hydrogen and oxygen, but it's not made up of cells. It's not a living thing. Rocks are not living. Fire is not a living thing. Buildings, I guess unless you live inside of a tree, Buildings are not living things. Your car is not a living thing. Air is not a living thing. There's more to categorizing what's living and not living than just whether or not it's made up of cells, though. So on the next page, we're going to look into what characteristics a living thing has, and all living things have in common. Another synonym, there's going to be a lot of words that mean very similar things, to a living thing is an organism. Organisms, this is the overarching term that we use to describe any living thing. Organisms can be broadly broken down into two categories. We have unicellular organisms and we have multicellular organisms. In this biology unit, you're going to come across a lot of words that have similar prefixes and suffixes. In fact, in science, there's a lot of that. The prefix uni, you can think of like a unicorn that has a single horn coming out of its forehead. Uni means one. So a unicellular organism has only one cell. Versus multi, I'm just going to highlight the word multi here, and then draw a little arrow off to the side, and I'm going to say that multi means many. At home, I have a multi-tool that has a screwdriver on it, a knife on it, pliers on it, has many different things in it. So a multicellular organism is made of many cells. We have some pictures here. You're probably familiar with the pictures on the right. There's some plants. Here's an adorable puppy. These are made of many cells. You can't overtly see the cells. You gotta use a microscope to zoom in and see the cells for these organisms. All animals are multicellular. So a cat is an example of an animal and a multicellular organism. Plants are multicellular, and fungi are also multicellular. Unicellular organisms, we're zooming way in on actually bacterial cells. This might be a streptococcus bacteria. Similar if you ever had like strep throat before or a strep infection, which is probably nasty, and you didn't like that, it might have been caused by these little guys here. This is a really zoomed in picture. You're not going to just see a picture like this by staring at your counter or staring at your skin trying to see bacteria. These are very small. But bacteria are a great example of unicellular organisms. There are also some unicellular eukaryotes called protists, but we might get into that later on. All of these organisms have the same 7 life processes in common. So all living things, remember living things we consider biotic. Anything that's biotic has these same 7 life processes in common. Now you're going to be tested on these. You need to know this list of how do you tell if something is living or not living. The M here, it stands for movement. Some people might use the word motility. You might see that. Motility. If you are looking up online later or watching like a crash course video on what makes a living thing, you might see the word motility. That just means movement. All living things have the ability to move, even plants. Now, you might think about some things that are alive, like a plant, and it's not going to run across your room. If you don't water it, it's not going to run to the sink to go get more water. But plants do move externally in response to stimulus. A plant will grow towards sunlight, for example. If you put a plant next to a window, it will lean towards the window so that its leaves get more sunlight. It's very slow movement, but it's movement. Some things don't move visually from the outside, but on the inside, they're moving things around. If you've ever seen a sea sponge before, they're just going to look like they're just sitting there on the bottom of the ocean, but inside they have very small cilia, these little kind of tentacle-like things that are moving around in order to move food in and out of the sponge. They're also going to move nutrients around their own body. That's a level of movement. Even if it's not running a race, it's still moving. That's something that all living things have in common. All living things are going to respire. When I say the word respire, you might think about respiration or breathing, and that's exactly right. Now, not all living things respire using oxygen. We have that written here. That's a little bit of a lie. Many things respire using oxygen, but there are organisms that respire using other chemicals instead of oxygen. But the thing that all organisms have in common is that they require energy. If you choose to take Bio 20 after Science 10, there's a whole unit about cellular respiration and photosynthesis, which are the processes by which cells get energy from chemicals or store energy in the form of chemicals. All living things require energy. Not all things require oxygen in order to make that energy, but you and most animals, all animals, require oxygen in order to do cellular respiration and get energy for their life processes. All living things sense the environment. The S here is for sense. That means they can detect changes in the surrounding. Now, you sense with your eyes and your ears and your nose and your tongue and by touching things. Those are your senses. You might not think that something like a plant has the same senses, but like I just said before, if you put a plant next to a window, it's going to bend its leaves toward the window. If you then spin that plant, then after a couple days, it will bend again towards the light in the window. That plant had to somehow sense the light in the environment in order to do that, in order to move towards the nutrients. All living things grow. That's what the G is for, meaning that they get bigger, that they mature throughout their lifespan. All living things reproduce, meaning that they make more of themselves. There's different methods to reproduction, but all living things will reproduce more of themselves. Trees and other plants will often make seeds. Many animals reproduce through eggs, or they reproduce sexually. Some bacteria, most bacteria reproduce what we call asexually, meaning that they can just divide themselves, but they make more organisms of the same type. All living things excrete. When you say excrete, that means that they get rid of a waste. As a byproduct of the chemical reactions that take place in order to sense, in order to move, in order to grow and reproduce, there's going to be waste materials that are generated that are not useful. So all living things will have to get rid of that waste. An example of you getting rid of that waste would be breathing out carbon dioxide. That's a waste material that you make from your respiration, and you get rid of that. Finally, there is nutrition, which means that all living things require a source of food. Some living things make their own food, but they require a chemical source of energy. These are all the things that all living things have in common. Non-living things will be missing at least one of these. So if you're thinking about, water from our last example, water can definitely move around, but the water molecules themselves don't reproduce. They don't change size. They don't grow. They don't excrete waste. They can't sense the environment. So even if a non-living thing has one of these, it doesn't mean it's a living thing. You need to have all of these in order to be classified as a living thing. Based on those 7 living things, take a look at these pictures and then decide where you think each of these items would fit. Would it fit in the biotic category or living? or the abiotic category, non-living. Go ahead and pause the video and try and classify these things as living or non-living. All right, so for this first picture here, we have a tree. A tree fits all the characteristics of a living thing. It's a plant. But also in this first picture, we have wind or air moving around. Wind is a non-living thing. A turtle is a living thing. Turtles reproduce, turtles eat food, turtles move around, turtles excrete waste. This is another plant here. This is I think it's a hydrangea, but I could be wrong. But a flower is an example of a living thing. Another plant. We got another plant in this picture as well as a bee. Insects are examples of living things. They're in fact animals. Rocks are non-living. Rocks, I mean, you could take a rock and you could split it, and then you end up with two rocks. And I guess you could call that a reproduction. But they don't grow then. You're being reductive there. And rocks don't respire. They don't require energy. And they don't move around on their own. Water is another example here, or clouds of a non-living thing. Clouds or rain, water, non-living thing. A dog is a living thing. And this last picture here, this is a picture of frost forming, probably on glass. So this is ice. That's a non-living thing as well. So I'm going to just put frost or ice here. Moving on. The next thing we're going to talk about is cell theory. We said that all living things are made up of cells. This is going to bring us to the idea of what a cell is. You're going to need to know the three parts to cell theory and describe some of the scientists involved in the production of cell theory. Before cell theory, there was an idea of spontaneous generation, which is the idea that living things, or biotic things, could arise from non-living matter. Here's a picture of a dust particle becoming a flea. So before the idea of a cell, people observed that if you left food, for example, out on the counter, it was all of a sudden full of maggots the next day. Or seemingly out of nowhere, fleas appeared in the house. And how did those fleas get there? Did they just come from dust? And there is this idea that non-living things would turn into living things. Now, we might see that as silly, but it was based on an idea of basically science of what people observed and realized. So if you cleaned your house, if you got the dust out of your house, then you had less fleas. So obviously the dust was what made the fleas appear, when in reality probably the fleas got there because they were on the back of a rat or something, and when you cleaned your house, then there wasn't rats there anymore, so the fleas didn't get into your house. But people weren't dumb, they just had a different idea of where living things came from. Then came along a scientist by the name of Louis Pasteur. You might have heard of pasteurization before, like pasteurized milk. Same guy. He disproved the theory of spontaneous generation, which is this idea that living things could generate from non-living matter. The word spontaneous here means kind of on its own, without prompting. This is the description of how he set up his experiment. So he set up experiment with this gooseneck flask. I have a picture of a gooseneck flask here. And he put some broth from a soup inside of this, sometimes called swan neck flask, sometimes called a gooseneck flask. And people had observed that if you leave soup out on the counter, then it's going to go bad. It's going to go rancid. Something's going to grow in it. So he took two flasks with the same broth, and they had S-shaped necks. And the broth was boiled in both flasks, to try and kill anything that was living there before. Then he broke the swan neck on one of the flasks, and he kept the swan neck on the other flask. Dust particles fell into both necks, but could not fight against gravity to move up the unbroken swan neck and into the flask. So in this one here, even though it's open to the atmosphere, then things land here, but they can't go against gravity to end up inside of the broth. In this flask, where he broke off the neck, things could fall straight into the broth. What he observed was that the broth in the broken flask became cloudy. You couldn't see right through it, meaning that something was growing inside there. There was probably some small unicellular organisms, perhaps some form of mold or a bacteria. But in this flask, even though it was open to the atmosphere, nothing grew. So that meant that life couldn't just generate from the air. Life couldn't just generate from the air or from the broth itself. It had to get in there. It didn't spontaneously generate. It had to fall into the flask. And if there was no path for things to go into the flask, then life wouldn't spontaneously generate. So it had to come from somewhere and go into the flask. Take a shot. What do you think the manipulated variable is in this experiment, and what's the responding variable? Remember that the manipulated variable is the thing that you change in this experiment. In this experiment, Pasteur was comparing a flask with a broken neck to a flask with an unbroken neck. So the manipulated variable is whether the neck of the flask was broken. And then what he was watching for, what he was measuring, that's our responding variable, he was watching for whether the broth became cloudy. Because this would tell you if microbes, it would indicate, that's what I'll write instead, indicate microbe growth. And if both became cloudy, then that means that microbes could just appear out of nowhere, or you'd have to set up a different experiment to see if there was a way to seal the flask to prevent them from getting in a different way. But what this one proved was that microbes had to come from somewhere. They didn't just appear in the broth on their own. Before we flip to the next page and talk about controlled variables, think about some things that Louis Pasteur would have to keep the same in order to make this a valid experiment. Controlled variables. Controlled variables are things that you have to keep the same, otherwise they're going to mess with your responding variable. So things that Pasteur had to keep the same, well, probably had to make sure that he had the same type of broth. I'll use blue for controlled variables. That's what I feel like using right now. Probably have to have the same type of broth. You have to leave the flasks in the same environment. You can keep thinking of some, but those are the two that I'm going to stick with right now. Because these are variables that if you didn't keep them the same between the two flasks, they could be the reason why the microbes grew in the one and not the other. Let's say, for example, he put chicken broth in the one flask that grew the microbes and beef broth in the flask that didn't grow the microbes. Who's to say that it wasn't actually the unbroken neck that kept the microbes out, but in fact it was the beef broth that kept the microbes out? Or if he put one flask in the fridge and one flask just at room temperature, who's to say it's not the temperature difference, the different environment, that changed the growth? What do you think that Pasteur found growing in the broth? I mean, if you leave food in the fridge for too long, or if you leave food on the counter for too long, you might see mold or bacteria. There's a couple things that could be here. Why did Pasteur boil both flasks of broth? If he didn't boil both flasks of broth, what do you think would have happened? I think that if he didn't boil both flasks of broth, that he would see growth in both flasks. He had to kill the microbes before the experiment started so that the experiment showed where microbes came from. It says here that the broth was boiled in both flasks. Right here it says these are the microbes being destroyed, all these little guys. After you boil the flask, they're dead. If you didn't boil the flasks, then there might have been microbes already in there, and both of these would have ended up cloudy. But by boiling the flasks, you can start with no microbes, and then your experiment figures out, okay, where do they come from? To make sure that there was no microbes at the start. I just realized that I keep using the word microbes, but I haven't explained what the word microbes mean. A microbe is just a microscopic organism. You can't see it with your eyes without the use of a microscope to zoom in really close. They're very small. What did Pasteur do to make the second flask different from the first flask? Well, that depends on which one you've labeled first or second. One had a broken neck. And both are technically open to the atmosphere, but we're going to say it was more open to the atmosphere. Explain how Pasteur disproved spontaneous generation using this experiment. Well, remember that spontaneous generation was the idea that living things could arise from non-living matter. Our broth here is our non-living matter. If spontaneous generation was true, then broth would be all that is required in order to make microbes appear. Because before you could observe, if I left the broth out on the counter, microbes appear. Where did they come from? Maybe they just came from the broth. But this experiment proves that the microbes didn't just come from the broth, because this flask here didn't grow any microbes. This flask here did grow microbes. So it showed that microbes must have come from the environment, not spontaneously generated from the broth. Microbes must have come from the environment, they didn't just spontaneously generate from broth. Okay, that's the end of Pasteur's experiment. Now we have a bunch of different scientists that you should know the name of. Robert Hooke. Robert Hooke was the person who is credited with coining the term cells. So when we say discovered cells, he's the one who called these little divisions of living things cells. And he did so by looking at cork. Cork is a type of plant. He used a microscope to zoom in on cork. This is what he saw. Now, he thought that all of these little boxes looked like little rooms, little cells. You can think of like a prison cell, but I think at this time, a cell was a room that somebody was in a monastery. Could be wrong on that. But these, he thought, looked like little rooms. These are little cells. So looking at cork under a microscope, he's like, okay, all these little divisions, I'm going to call them cells. Anton van Leeuwenhoek is considered the father of microscopy because of the advances he made in microscope design and use. And he helped develop the compound microscope and really turn it into the sort of microscopes that we use today. There's two German scientists in the mid-1800s who were responsible for initiating cell theory. What cell theory actually is, we'll get to on the next page right away. Schleiden was studying plant cells, and he observed that every plant cell contained a nucleus and that each new cell developed from the nucleus of another cell. So that's going to be two important parts of cell theory. Everything, all these plants have cells, and those cells come from other cells. Schwann, who did a lot of research in cells, and there's a cell named after him in your body called a Schwann cell, Schwann found cells in animal tissue. So Robert Hooke was looking at cells in plants. So was Schleiden looking at cells in plants. Schwann found cells in animals. So now both of these have cells. Cells are the basic structural, functional, and biological unit of all known organisms. Here is a list, a little timeline of when these things happen. So Robert Hooke, like 200 years before Schwann and Schleiden, is the one who is naming cells in these cork cells that he's looking at. And then, so he must have been looking under some form of microscope. Anton von Leeuwenhoek got even better microscope designs. He was able to see more types of cells because he could zoom in even more. So these are the important ones to tell here, as well as Schwann. Schwann was looking at animal cells, and then Schleiden, who was looking at plant cells. There's other scientists on here as well. That's going to bring us to where we're going to end off today, which is cell theory. And then cell theory will bring us to our next lesson where we talk about what actually is in a cell. Cell theory has 3 main points. First point, all organisms are composed of cells, meaning all living things. Remember, this word organism is biotic. All living things. All living things are composed or made up of cells. And the cell is the smallest functional unit of life. What that means is if you go smaller than a cell, then it's not living anymore. The cell is the smallest you can go for a living thing. The cell is the smallest functional unit of life according to cell theory. All organisms are made of cells. The cell is the smallest functional unit of life. You can have one cell and you're still living, but you need at least one cell. And all cells have to come from other cells. All cells come from other cells, meaning that they don't spontaneously generate. An existing cell has to replicate to form a new cell. There's a bunch of good review questions here. I would encourage you to take a look at them. And then in our next lesson, we'll talk about what's actually inside of a cell and what those different pieces do.